delivering it today. Patty is not only a friend of mine, a longtime friend and colleague, but also a trusted leader of the advocate community. Patty's also a consultant for CISN, and we are really pleased to have her deliver this webinar because her knowledge of clinical trials is extensive, and I'm sure you'll learn a lot today. So Patty will be delivering the webinar. As you can see, I can mute you here, and it helps if you mute your phone, and we'll unmute at the end, like always. Today's webinar is entitled Clinical Trials 201. We do have a 101 for very beginners. So if you know people, if you have friends or advocates that are brand new and really need a very, very beginning course, we do have a Clinical Trials 101. This one is more in depth, and that's the one we'll be delivering today. The presentation will take about an hour. There'll be questions at the end. The presentation is also being recorded. You can raise your hand at the end if you're talking and Patty's not answering you or I'm not answering you, then please feel free to type in a question. Patty doesn't see that screen, but I do, and I'll convey the question back over to Patty. So our mission, again, is to build bridges between all stakeholders, and we use the internet a lot to do that. The curriculum developers, the web content for Clinical Trials 101 and 201 was done with Jane Perlmutter and myself, but the PowerPoint content and the webinar today was between myself and Patty. So welcome, and feel free to ask questions at the end. But remember, we're not doctors, we're not giving advice, we're not making recommendations, we're just providing information. So Patty, please feel free to take over. Thanks so much.
Okay, thanks Peggy. That was a nice introduction. So welcome to Clinical Trials 201. So the learning objectives for this training are to better understand the whole cancer research process, as well as to better understand clinical trials. And so some of the information might be a little bit technical for some people, but it might be that I know some people online might actually have more experience than I in this. And so if you think of something as we're going through the presentation that you want to add at the end, please feel free to do so. Because you might have, wisdom that others might benefit from as well. So this training is for us to train each other as well. So these are the objectives of the training. And I think I'll see if I can go to the next slide. I'm not sure if it's going. No. Okay. If you can do it, Peggy, that would be great. Okay, so this is the overview of what we'll discuss today. We'll discuss the basic research process, basic translational and clinical, as well as clinical trials more in depth. So we will learn to then move on to clinical trials and how this method is required by the FDA to test any new treatments before they're used in humans. So the research process. So during this section, we'll provide a bit of background on the research process used to develop new cancer treatments. We'll try to capture some of the excitement, but also some of the disappointments, or like I like to say, the realities of conducting research specifically in biological systems like cancer. It is quite difficult to do. So all research is based on the following, the scientific method. You may remember learning this at elementary school, middle school, high school, and it's still followed today. It's your basic scientific method. And most of us follow this in our basic decision making every day. We observe, we hypothesize, we test, and we conclude. And I read a really good example the other day on, you observe and you try to gather as much information as you can. And so you might make the observation in your garden that some of your tomato plants are growing bigger than other tomato plants, and you wonder why that is. And so you kind of have to observe the situation and gather information. And there are several different things that you notice, like water content, where you got the plant from, the amount of sun that it gets, and then you kind of make a hypothesis based on your observations. And your hypothesis is like, well, what do you think is really happening? And so you might say, well, I think it's the sun, you know, the amount of sun. And then you kind of say, well, how could I test that? So if you have the same plant from the same place, the same amount of water to each plant, but you just vary the sun that each plant gets, then you can test that hypothesis. And then you conclude based on your thing, if the plant in the sun grew much bigger than the plant in the shade, then you could say, yes, it is the sun. So you kind of do that every day in what you do, and that's kind of what scientists do too as they move through the research process. So next slide. I don't know if mine is doing it. So in recent years, cancer research has increasingly required teams of scientists from many different disciplines. And I think we're seeing this really in cancer a lot, with physical scientists and other scientists joining the MELD. And this slide provides you with the 4 basic categories of research. So you have your basic research, which is your basic advancement of knowledge and how things work. You have translational research, which is moving this basic knowledge from the bench in the basic realm into animal testing, and might even include tissue testing. And then you have your clinical research, which takes that translational research and moves it into people. And this is where is usually called clinical trials. And then you also have epidemiology, where you gather information from the large groups and draw conclusions from that. And you can see here that each category leads to the next. So without basic research, you would never get clinical research. So it's really important to keep that in mind as we go through the importance of basic research going through this. So the most important thing to know about cancer research is that it is the road path to new treatments. New treatments are needed to improve patient outcome and lessen side effects. Research costs are very high and take a long time. We'll discuss this a little more later. And government research focuses on the public good, which drugs work better, whereas industry research is focused on the return of investment. Both types are needed to contribute to the advancement of science and the advancement of new therapies for cancer. So what is the reality of basic research? We've just discussed a little bit about the research process and its importance to lead us to clinical trials, but we'll now discuss in greater detail each step beginning with basic or bench science. And this research is the furthest from the patient. So remember the research arrow, it's the furthest from, translating into patients, but it's the place basic understanding of how cancer cells work is learned. Like all beginning efforts, there are many failures as information is learned, but it's not really a failure because you are learning something. So you are learning new knowledge that is true and reproducible. So you'll always be learning and moving things forward. And in reality, big breakthroughs are usually done through serendipity, and that means by chance. It's not something that you planned on happening, but something that you saw happen from your observation. And there's a lot of new technologies out there too that are helping us move this basic research forward a little bit faster than it used to move if you look 25 years ago. So there is a lot of really good science moving forward right now. So translational research. So translational research science also has more failures than successes. But remember, we're still going through the process of learning. And so every time we go through a process, we learn something. So we're taking baby steps through the process. One of the successes developed, many that work in mouse models, cannot necessarily be used in people because they are either too toxic or not effective. They could be effective in mice, but not necessarily in people. But we still have to do the research to bring things forward. So even if something passes this step of translational research in mice, it still doesn't mean it's going to make it to the clinic. So remember, the point of clinical research is not only to identify the research that should go into clinical trials, but also to identify those things that should not go into clinical trials. So this is a nice place where you also pick up the ones that you should not bring forward because they are too toxic or they're not effective even in mice. So that seems like that's one of the hardest things to do, is to put a lot of investment into a project, bring it through, to your mouse models and your different things, and then find out that you can't bring it forward. It's really hard to stop something, but it really has to be done because we really can't bring everything into human clinical trials. Next slide. Okay. So the reality of clinical research, and clinical research is difficult. This is the research that tests the new treatments that are successful in mice in people. These are called clinical trials. The rest of this training will focus on clinical trials and help you understand them a little bit better. We hope you now understand the research process that leads up to clinical trials, and understand the importance of that research that leads you up to a clinical trial. The path to approval is a process which will further identify winners and losers in a way that is safe to put them into patients, because those first-in-human trials are very important that you have some safety information before you go into humans. But still, once you go into this clinical trial arena, there's still time to stop something that is not going to benefit patients. It's not, so there are still some failures. Not everything leads to a treatment, and I think that's important to know as well. And that's also hard for people to do, to stop something that is minimally effective and maybe too toxic for patients. So I don't know if I'm moving the slides or not. Yeah. I'm not sure if Peggy's doing it for me. So this slide is another version of the arrow that we saw earlier, that during the basic phase, biologists work in the laboratory. I don't know if you can see my cursor. Biologists work in the laboratory and may study cell cultures, petri dishes. Chemists may work to develop molecules with specific structures and functions that provide better treatments. And this is all done in the basic realm. During the translation phase, new ideas are often tested in live laboratory animals, often in mice, and this can also include testing in human tissue. And the clinical trial phase is where we test it in patients, and that's what we're going to talk about now going forward. So we'll now focus on clinical trials so you'll have a better idea. So write down any questions that you have. You can type them into the chat as we go, or just wait till the end to ask any questions, but be sure to write them down as we go through this. So what we're going to discuss right now are, what are clinical trials, what are the types of clinical trials, the phases of clinical trials, different components of the clinical trial, the protections that are in place, barriers to participation, the big accrual issue that we have, and who conducts clinical trials. Next slide. So definitions of clinical trials. So a clinical trial are research studies involving people. They try to answer scientific questions and find better ways to prevent, diagnose, or treat cancer. All advances that we now enjoy, whether in cancer or other diseases, must first be tested in clinical trials. So very, very important. So why are clinical trials important? So because cancer is so prevalent. So 1 in 2 men and 1 in 3 women will get cancer sometime in their lifetime. So we need very good treatments. Clinical trials test new treatments before they can be approved by FDA and used in patients. And we should always encourage people to ask about clinical trials when they're diagnosed with cancer, because those that are eligible should always be asked to participate in a clinical trial. So whether you or someone you know is diagnosed, it's always a good idea to ask your doctor about clinical trials. So I love this slide. So many inventions interventions work in animal models but not in people. Again, the FDA will not approve any new treatment before it's tested in people. That is because that even if it works in mice, it may not work in people. So even when you see in the news and in the press, you know, these big breakthroughs, and then you read the fine print and you read that, yes, this breakthrough was in this mouse model, tested in this mouse model, and the mice did great, it still needs to be tested in humans, and not everything that works in mice works in humans. So now we'll move on to the types of clinical trials. So most people think that only new drugs are tested in clinical trials, but that's not really true. We also test new surgical and radiology procedures, also must be proven in clinical trials. Prevention, screening, follow-up strategies, and survivorship are also tested in clinical trials. Sometimes you'll test dose scheduling and sequencing of drugs, comparing two different drugs. Those are all done in clinical trials. So it's not just an intervention or not an intervention, a drug. It's sometimes a lot of other things. So there's a big array of different types of trials out there. So the phases of clinical trials. So clinical trials are performed by going through a series of steps or phases. Usually there are 4 phases. So biostatisticians and others are always improving the process to answer the questions more quickly, but we'll discuss here first the usual 4 and several new designs as well. So always ask your doctor when you volunteer for a trial which phase of the trial you are entering, because the phase does matter as far as risk and benefit. And whereas the phase 1 or phase 0s have the highest risk, the phase 3s usually have the least amount of risk and the most likelihood of benefit. So we'll go over the typical 4. We'll go over some ideas that are changing with more targeted therapies, and then of course adaptive design is something that's on the forefront as well. So we'll go through the phase 0 study. This is used usually with targeted therapies when they're first being tested to see if the drug actually hits the target. It also looks at the effect on cancer cells and how well the body is metabolizing it. This saves a lot of time and money. No sense in wasting time and money on a drug that doesn't hit the target. But not every drug will go through phase 0 testing. It's more for the biological treatments. And the important thing about this one is that there's no therapeutic intent, because there's a suboptimum amount of drug that goes in. It's not the treatment dose, it's a lower dose. They're just looking for the effect on the target. So there's absolutely no benefit, but there still may be risk, even though it is a low dosage, and that risk is very unknown. So this is a very select group that may go through this, and not every drug will go through a phase 0 trial. So phase 1 is where most drugs begin. So most drugs don't do the phase 0 trials, but every drug goes through a phase 1 trial. So for each of the phases, we'll show the purpose, the number of people typically involved, the risk relatively, and the time it usually takes to complete the trial. So this phase 1, is not designed to test the drug, how effective the drug is, but rather how much the drug can be given before it becomes toxic with too many side effects. And this is kind of changing in the world of phase 1 trials these days, but this is your typical where you look for the dose-limiting toxicity, you put the least number of patients on before you go up in dose, that you know that it's safe with 3 patients before you put on 3 more patients. It's the least number of people, only about 30 people typically, but I can tell you now some of the phase 1s are going up and up and up in the number of people. This is the most risky trial to be on as a patient, and the benefit is really unknown, and they're not even measuring the benefit usually in phase 1 trials, whereas now they tend to look at that, but they're not powered to look at that. And the time is not the same in all cancers. So this is where all drugs start, and is usually the safety part of it. So phase 2 trials is the next step, is called phase 2, and it's important to understand all of these phases and what their purpose is, as well as the risk, and the number of people to reach the answer in phase 2s is determined by a biostatistician. It's usually 100 people or so, and this can usually be done in a few centers, and it takes a little bit longer than a phase 1, usually up to 2 years to accrue. And this is also where they continue to monitor the safety, but this is where they start looking at effectiveness and look at different endpoints and start to see if the drug is effective against a certain cancer. So phase 2 trials are really, I think, are the nuts and bolts of a lot of what's going on, and this is the area where I think that the go, no-go decisions really need to be made before they go up to a phase 3. But I think this is where you can gather quite a bit of information as well. So this phase 3, is where the new drugs are tested against standard of care. And this is when it's referred to the FDA to see if it's a newer, better treatment than the standard of care. It requires many people, takes a long time, the longest amount of time, so you usually need multiple institutions, lots of people, lots of time. It has the least amount of risk because it's already been through the phase 1 and phase 2, but still is experimental, so you still don't know if the treatment is really better or worse. And I think that this is changing in the future with these really exceptional drugs that have a lot of benefit. You know, this is becoming a little more difficult to do. But I think that, you know, this is where the crux of the FDA approval happens. And, you know, nobody ever gets nothing. It's against the standard of care. Different endpoints are used. And these can take a pretty long time before they're not only completed, but then reported out. And then there's another phase 4 trials, that's not really a clinical trial. Actually, this is where people are not entered into this phase, but rather it's an ongoing process of gathering information after a drug has received FDA approval and is now being used by the general cancer population. Important information can be learned about effectiveness and side effects as huge numbers of patients start using the drugs. And this is really important, especially for long-term side effects, and because you know, wider range of people are using the drugs. So people with comorbidities are using the drugs, and people that are sicker than those that go on trials are using the drugs. So this is where you can really get that information on not only the effectiveness but toxicity of the drug. And so some people really like pictures. I really like pictures, so I really like this slide, and it's always helpful to have pictures. So this is a summary of the 4 phases of the trial, including so it shows you the information gained from each trial, and it includes the purpose and risk of each trial and how many people take part, the dollar amounts to conduct, and approximately how long it takes to conduct the trials. Remember, clinical trials are designed to test new drugs and treatments, not to treat those in the clinical trial. Though you may end up benefiting, it is still a study to test a treatment. So, however, things are changing, and the world of clinical trials, due to several factors. Due to many of our trials now are more science-driven, and also I think there's a real push for the efficiency of drug development and to get that done more quickly. And so some of the lines have been blurred between all of the phases of clinical trials. So one of the biggest things that have changed is the phase 1 trials. And phase 1 trials are bigger because they go into expansion cohorts. So you'll go from a phase 1 to a phase 1B in one protocol. in some cases, where you look at the toxicity in the phase 1, but then you do expansions into different combinations with the standard of therapy drug, different tumor types, different molecular subtypes, different doses and scheduling. So it's reported that one pharmaceutical phase 1 trial actually got up to 1,200 participants because they tested it in so many different tumor types. And these different expansion cohorts can range from 10 to 180 participants. So, you know, these are kind of changing where they're looking at different things in phase 1s than they used to, because I think we're kind of trying to get more information earlier, as early as possible. And so we're also seeing combinations of all of the phases of trials. They're very common now. Sometimes they're called run-in trials, where the phase 3 portion is an expansion of the phase 2 study. So the same protocol and same participants can be used in the phase 2 and 3. So consenting as a patient advocate on some of these protocols and looking at some of these consent forms, I think consenting patients to these trials, we need to be aware of how that consent form reads, because a phase 1 trial and phase 2 are very different in the consenting process, and what you tell the patient and what you know about the drug, because you're definitely going to know more in the phase 2 phase of the trial than the phase 1. So, you know, how you consent that patient is important to us as patient advocates. But there are a lot of advantages to doing trials this way, and one is that you can use one protocol and pretty much get 2 trials done. Less total patients can be used because you're using the patients that consented to the phase 2 to be in the expansion of the phase 3. So I think there's a lot of benefits to it, and so I think we're seeing more and more of this happening as we go along. So as we go along, this is just a few things that I've noticed, and I'm sure others have noticed other things that are changing coming down the pike. I'm moving it. Oops. Okay, I think we go back. There we go. Okay, so the trial protocol. So every clinical trial has a protocol. And so we're going to switch gears now and talk about this document and how clinical trials are designed and conducted. And this protocol is a very important document. It outlines the science behind the study, the purpose, the risk, and the benefits for future patients. And it's very important that every site follows the exact same steps so that all the data can be used. So it's important that this document is very good and goes to all the sites. And this includes a lot of things, and one of the things it does include is the consent form that explains the study and other options and lays out risks, and needs to be understood by the patient. So what will you see in the clinical trial protocol, and what should you look at? So every clinical trial has the same required categories that are spelled out to ensure that every study is conducted the exact same way. And we'll discuss some of this in the next slides. Also, every doctor in the study site enrolling patients into the specific trial must follow the details in that protocol to ensure the data can be compared across sites. It needs to be done the exact same way. So we'll talk about eligibility, who can join, endpoints, what they are, trial designs, randomizations, blinding, and the consent form, including risk, benefit, and protections. So we'll go into each of those separately. So eligibility, again, after explaining the science and the goals and the risk and the benefit for the trial, an important component of every protocol is eligibility. And these are detailed specifics about the enrollees must meet. So inclusion criteria are things that patients must have, and exclusion criteria are those things that patients cannot have. So it's important for us to look at and to question when they say that this is an inclusion criteria, ask if that really is necessary, because the more patients we can get on trials, the better, and if they're making them too narrow, we really don't want that. But in some cases, like health status criteria, it might be some criteria that they don't want a patient to have because of the drug that they're actually giving within the trial. So some are definitely necessary and some are not. So we need to kind of look through those, what the inclusion and exclusion criteria are. So endpoints. So endpoints could be a whole webinar unto itself, I think. And the more you look into endpoints, I think the more confusing it gets. But it's a critical component of every study. These are set before the study starts. It's very important that they're set and designed to show if the drug is effective at killing the cancer cells and extending survival. Here are some of the most often used endpoints. Although extending survival is the best, others are often used because they can be met in a shorter time period. So endpoints matter. For example, objective response rate is used a lot and includes partial and complete responses, but not necessarily stable disease. So progression-free survival is another one that's used a lot as opposed to time to progression, which sound a lot alike, but progression-free survival includes death, whereas time to progression does not. So there's these little nuances with each endpoint. So it's important as advocates when there's an endpoint in a trial to kind of look up and know what that means. So if you're unsure about it, you can look it up and find out, because it is important and it will be important to patients going on that trial. If stable disease is important and a good outcome for the patient population, then objective response rate might not be the one that you want to use. So trial design. So there's a lot of different trial designs out there and some alternative designs being proposed, which I think are really interesting in moving this area forward much faster. One of the classic designs is the randomized controlled trial, which you'll see referred to as RCT. It's the gold standard because this is where the treatment is compared to the standard of care. And this is the best way to see which is better. Patients do not like this because they don't know what they're getting, if they're getting the drug or the standard of care, if they're randomized, which can be a barrier to accrual, especially if there's hype in the media about the therapy being really effective. It's really hard to randomize patients in this way, but this is the gold standard and this is what we try to do because each trial should be equipoise and where you don't know the outcome. So in order to alleviate the placebo effect, you do the randomization. And this is your typical way of doing things. So the next thing we'll talk about, an even better design, is to group people ahead of time by a certain characteristic, like a genomic KRAS plus or minus, or behavior like smoking or non-smoking first, and this is called stratification, and then you randomize after that to the agent or the control. Selection on a biomarker is becoming more normal, but also might introduce some unevenness if the marker that you're stratifying for is, there's a lot more of KRAS negative than positive, then you're going to be skewed in your stratification at the onset. So this can be a little difficult to do. But a lot of times what trials do is they do a randomized controlled trial, but they plan it in such a way that they gather the information and they try to get enough patients so that they can stratify afterwards, which is usually a different way of doing the stratification after the randomization. This is common, takes a broad group that have the different biomarkers but doesn't look at it until after they're randomized. You still don't usually know if you have enough, and usually this stratification factors are usually secondary or exploratory aims of the study, not primary aims like it would be if the stratification was up front. But you still need to make sure up front, if you're going to stratify at the end, that you collect the information before the trial gets started, because if you don't plan on that, you can't do the stratification afterwards. So it's very important to know what you're looking at before you start the trial so everything is done in a common way. And sometimes I can do it and sometimes I can't. Okay, so then go back one. There. Okay, so blinding. Blinding is a term used when the patient does not know whether they received the new drug or the standard of care. Double blinding is when the doctor and patient don't know if the patient got the intervention or the standard of care. And blinding is important because it relieves all the bias, the placebo effect, preconceived ideas about the agent. But we do have some agents out there now that have very specific side effects that are very different from the standard of care. So a lot of times, even though it's blinded, I think the doctors kind of realize which patients they have that are on the intervention and not. But this is typically done so that that you don't know, and this is how most of them are done these days, which is the best way to get the best information out. So now we'll go into some new trial designs. And so we've heard about these on some recent calls with the NCI about some new trials. We'll go into the umbrella and the basket design because this is the way that things are going, and these are some new type of trials out there. The umbrella trials are genomically driven trials, but in one tumor type. So you have one cancer type, like lung cancer. There's two trials ongoing now within the NCTN, the Alchemist trial and the LungMap trial. So, or the Alchemist is in the NCTN. So these are some really good examples. So the tumor, you get the molecular finger footprint of the tumor of each person called the genotype, and then you assign them to a drug that is targeted to that tumor and that mutation. So a mutation is found, so you find mutation 1, they get drug A. You find mutation 2, they get drug B, and then this usually comes with a standard of care arm. So if the participant doesn't have a mutation, they go into the no mutation arm, and then they get standard of care. In some cases with the LungMap, I think they were doing another drug that was very promising in the control arm. So I think these are very good trials, but they're only one cancer type. And they're called umbrella trials. I'm not really sure how they come up with these names, but I guess it looks like an umbrella. And another new one is the basket trials. So these are also genomically driven trials for all tumor types. So these are agnostic to the body part from which the tumor came. So this is called the basket trial. So every person in the trial has the same mutation, but they might have any type of cancer. So this allows for a small number of people from any one cancer type to possibly benefit from a drug that targets that genomic mutation. So any cancer type can enter the trial. This is how the NCI Match trial is set up, and that's the new trial through the NCI that should be open by now because it's August, but I'm not sure it is. And so this is all cancer types. So if you have, a breast cancer, lung cancer, a pancreatic cancer that all have this mutation 1, they get into this little bucket and they all get drug A. So you're kind of running several little mini trials within this mutation set in all different cancer types. And so the Match trial is set up to look for signals. So they're thinking, if you have this mutation and you have, colon cancer, and we have a few patients on there and we start seeing a signal, then that can be expanded to a true phase 2 trial in that population. But this is a way where they can just kind of get all comers coming in, and as they identify mutations, they can parse them out. This does not have a control arm, so they have to screen a lot of patients to get enough patients to fill all the arms. And I think it's going to be, I've got this etc. there, it's going to be up to 20, I think, buckets for the Match trial. And they'll be taking buckets out and putting new buckets in as the trial progresses. But this is a new way of looking at cancer, not as body part cancer, but as different mutation types. So this slide kind of shows the difficulties with, how we develop drugs and the drug treatments today. And in most cases, we don't know who needs or who will respond to a given drug, even though we know that most cancers have several or more subtypes. And this is not a good approach because we know, even if you have a HER2 overexpressing breast cancer, you're not necessarily going to respond to Herceptin. So, or respond for a long time. So, it's still kind of like a whack-a-mole that you just kind of try and try again. And this is just not a good approach. But we think things are improving, and I think the new trials that we're doing, trying to really understand the differences in the tumors and treating them appropriately is really going to get us forward. And we're doing the smarter trials and doing the new ideas and the adaptive design, find out early that something is not working and move on rather than keep banging your head against the wall. But I think that things are changing, and, I hope that with these new trial designs and new things going on that we'll get there faster and we'll be smarter about what drugs we give, and it won't be such a whack-a-mole effect. So now we'll move on to an important part of the protocol. And so every protocol and clinical trial is the patient consent form that a patient must sign before they enter a trial. All aspects of the trial, including purpose, benefit, risk, confidentiality, financial responsibility, and how it compares to standard of care, must be explained. Patients should be given the form to take home and have all their questions answered and understand that they are entering a research experiment. So I think it's really important that, we as advocates, really look at these consent forms, because these are forms that they're going to bring home and read later, read with their family. But also I think, we also have to understand what the process is at the different institutions that we volunteer at. With the consent form, actually it's a consent process. So I think that it's up to us really to help with that as well. So statistics. So this is also another topic that could go for an entire webinar, and I was trying to think of easier ways to say anything, but there's really no easy way to do this without like a whole another set of 10 slides. So we talked about endpoints earlier, and statisticians are part of the clinical trial process, and they design and track endpoints and goals and make sure they're met and use these statistical models. So we hear about p-value a lot. So the p-value is important as it documents if the result is real or if it could happen by chance. So, if something happens by chance, it's not real, but if it's real, then it won't happen by chance. So the usual breakpoint is 0.05 for a result to be deemed real. So, the likelihood of it being done by chance or not is basically what the p-value is, and it's kind of backwards, so I won't even go into that. But statistics in trial is complicated. And so your confidence interval is another value that's very important. So it's not only the p-value. So even if you have a p-value of less than 0.05, so you're pretty sure it's not by chance that you got this value. It's a real value that's different from your null hypothesis. Your confidence interval is very, very important too, and this shows the variability of the study range. So it'll show what each person on the subject on the trial fell within that line of values. And so, if there's a very, very wide range, then you have less confidence in your p-value. If there's a very narrow range of your confidence interval, then you have more confidence in your p-value. So even if you have a p-value that's significant, if your confidence interval is huge and crosses the barrier of the null, then it really isn't true. So you really have to look at that as well. So it's kind of a combination of those two things. Sample size is something very important. You need enough patients to get the answer, and your statistician usually determines that up front, and that's very, very important. So when we have these trials that lag an accrual and can't complete accrual, it's very important to know what you can and cannot determine with that substandard sample size, and your statistician is the one that can tell you that. Relative risk is another one that we look at, which is the likelihood that a cancer will occur within a specific time frame, and it's a comparative of one group versus another group. So there's a lot of other terms also, but this is just kind of some of the major ones. But you could do a whole webinar on statistics, which maybe that's what we'll do in the future, who knows. It's an important part of the trial for sure. So this is an area that's really critical, that all trial results are published, not only published, but published quickly, and that they include the negative results as well as the positive results. Doctors in the community practices base what they do on what's published and determine the best treatment for any given disease. So unless it's published, it'll never make it to the community practicing oncologist. I almost said veterinarians there. They find this information from publications and discussions about these publications at research meetings. So unless they're presented or published, the community doesn't find out. Unfortunately, publishing takes time and money, and there's often no incentive to do this, especially for negative results. Trial results are supposed to be uploaded to clinicaltrials.gov, but the format is very, very awkward, and timeliness is questionable, especially in academic institutions. But it's a start. But I think there is a big push right now to get research study results published, not only in the scientific community, so community doctors can have access to it, but also in the patient community, so patients can have access to the information as well. But still, it's really going to be hard to get anybody interested in reading about negative results, but if we don't publish them, then we'll just repeat them. So it's very important to get that information out. So protections. So it's always important when you're reading a clinical trial that there are protections in place, and safety does come first. So we can't put anything into patients without knowing, having these protections in place. So there are federal laws out there and local protections that require close monitoring. So the consent form has the complete disclosures of all the risk, but I think that, just knowing that there's federal laws out there, the National Commission for the Protection of Human Subjects and the National Research Act, and also before any study gets started, it is approved by an institutional review board, and they look specifically at patient protection. And so you cannot open a study unless it goes through an IRB. Institutional review board. So every patient should know that, and everybody should know that. And then during the study, and when I read protocols and read consents, I always look for the data safety monitoring board, that there is a data safety monitoring board, that they're looking at it at reasonable amounts of time. They look for not only toxicity but futility of the treatments. And so, these are very important parts, but in the data safety monitoring, it's very important to define what they're looking at up front and what you constitute too toxic or your futility rules during the study. So there's a lot of things to look at, but I think it's all in the benefit of the patient, and there's a lot in place. So what are some of the barriers to participation? So each clinical trial is different, but each have their own barriers. And these barriers are both from the doctor and patient perspective. So this slide tells you the barriers that the doctor might have. but others exist as well. So these are just a few. So they might just be unwilling to put patients on. They might not believe that this treatment would work and believe the standard of therapy is better. So a lot of times it's getting the clinician on board as well. And if you can't get clinicians on board, then you might not. I said there might be a problem with the trial. So I think that we really need to think about that as trials go forward as well. A lot of times clinical trials cost more for the clinician to do and their practice to do, and they won't do it because of that. And so we need to keep that in mind. But those are some physician barriers. There's also patient barriers. So patients might just not be aware of clinical trials because the number one reason for patients participating in clinical trials is that their physician asks them to participate. So if their physician doesn't ask them, then they don't know, they'll never get in. Some just have lack of access to trial, fear of suspicion of risk. They feel like they might be too risky or they're taking on too much risk. They might have personal obstacles. Sometimes there's more visits involved, and they might not have transportation, and that might be a real issue for them. Some might have insurance costs. Sometimes there are heftier costs to being in trials. And some just can't make the decision. You know, when you're diagnosed with cancer, there's so many decisions to make. And, you know, if you have to make this one on top of all of your other decisions, that might just be too much for you to handle. And so you might just decide not to do it because of that. So this is a very important slide that Peggy does pretty much in all of her presentations, but I think it's really, really insightful that, you know, because it captures everything that happens. So barriers to participation. It's complicated. The consent form looks like a legal document that's 20, sometimes 30 pages long, you know, and that can be a barrier. There's often few graphic handouts. I've seen very few graphic handouts. Sometimes I see a graphic handout that's still not very helpful. So I think not only few, but not well developed. Incomplete staff training, you know, participation is a process. And it's a process involving people, including the staff and the participant. And so training is an issue. Not enough time. You know, the clinic visits are very short sometimes, you know, and it takes time to consent a patient to a trial. Cultural differences. Some cultures just don't. You need to be aware of those cultural differences, and that, you know, you might need to state things a little differently, or have somebody talk to someone from the same culture and get them on board. Low literacy rates. The complicated consent form is very hard for someone who doesn't read English to read, and actually sometimes I read them and I can't understand them. So low literacy rates. Different learning styles. I like pictures, and there's usually no pictures. Some people like the verbal, some people like the written and like to bring it home and really study it. So everybody has a different style. So it's a different, so it's kind of the perfect storm for non-participation. So can we have more graphic handouts? Can we have more patient-centered staff training? Can we allow more time for participation? Can we address cultural differences in health literacy and learning styles and make this process a better process for everyone? And then we'll get more people on trials. So what are the benefits to participating and enrolling in clinical trials? This is a volunteer choice, and anyone can drop out or be at any time and begin standard of care. So it's not a thing that you can't change over time, and I think a lot of participants don't realize that. And again, it's important that people understand that they are entering an experiment that may be better than, but it might be the same as, or it might be worse than standard of care. So it is still an experiment, but you still might receive, you know, you might receive more follow-up, you know, and you might receive benefit. And I think some of us have been in clinical trials where we don't know if that's why we're still here, or that's, you know, did that for us, but we hope that that's why it is, but you never know. But it might improve the standard of care, and it might move us forward in this specific area of drug development. And so, you kind of have that altruistic part of it too. And, you do receive, pretty close supervision. You're seen pretty often. You can receive more scanning, things like that sometimes. So, sometimes it is a benefit to be in a trial. But as we talk about benefit, but there's also risks of being in trials. So, we like to talk about the benefits, but there are these risks, and some of these are unknown side effects. So these are new drugs going into people, sometimes for the first time if it's a phase 1. And so sometimes it's just unknown. There's therapeutic misconception. Some people think that if I go in a trial, it's definitely going to work all the time, and that's not the case. We don't know if it's going to work better or not. Sometimes there's extra costs. Sometimes extra procedures are good for some. Some like the extra procedures or extra scanning, and some don't because it increases anxiety, increases the office cost, and increases your copays. And sometimes there's just unknown risks for participating in clinical trials because it is different than just going to the doctor for your standard of care. So it's critical that patients have all their questions answered before they sign the consent form and enter a clinical trial. Will there be extra visits? Tell me about the study, what the study's about, who put the study together, where are the trials being conducted, what will I get out of the study, what are the risks, how long will I be in the study? There's a lot of different questions, and I think you can find questions to ask your doctor about clinical trials online as well, which is really good because I think that there are a lot of unknowns sometimes in clinical trials. It should be spelled out in the consent form, but sometimes these are things that you need to really ask the doctor. So who does conduct clinical trials? There are several different groups that conduct clinical trials. Here's a few. The National Cancer Institute, through their grant funding mechanisms, are ones like that. They fund clinical trials. The NCTN that we're all involved with conducts clinical trials. There's some consortia out there that also conduct clinical trials. And of course your academic centers, community hospitals, and your healthcare provider, and of course pharmaceutical companies also conduct clinical trials. Next slide. Okay, so how do you find a clinical trial? Well, most times it's online. You can also ask your doctor about clinical trials. Here's some standard places to go: clinicaltrials.gov, cancertrialshelp.org. Also, some nonprofits have their own sites. The Leukemia Lymphoma Society has a nice search engine site. Breastcancertrials.org has a nice site. So different cancers also have their own sites, and your doctor is also good source of information, and the institution that you go to be treated is a good place to go as well. They each have their own clinical trials listed. So next slide. So the goals of clinical trials, let's click through there. So you can see the goals of the public and industry trials are different. Let me click through the next one. Get the public sector up there. So you can see that they're a little bit different, and I think that you can see that we need both publicly funded trials as well as commercially sponsored trials. There's a role for both. The commercial sponsors, that's your drug registration trials, and that's how we get drugs to the market. They do label extensions, expand their market share, but they're also they need to create shareholder value in the end. Whereas publicly funded trials, the ones that we're involved with, the NCTN, we can look at optimizing treatments, we can look at label extension as well, we can create new knowledge, and we're really after improving public health. And we can do the ones that compare one drug to another from different companies as well. So there is a role for commercially sponsored trials as well as publicly sponsored trials. slide. I'm not sure if I'm doing it yet either. So why do we need publicly funded trials? When we say publicly funded, we mean government funded. When funding for research goes down, this includes all stages of clinical trials. Industry, of course, is trying to develop better treatments, but they're also focused on their shareholders. Publicly funded trials are concerned mostly about bettering public health outcomes. They compare different drugs, they test to see which dose is better, which sequence is given better. So there's a lot of things that we can do in publicly funded trials. So, we need to be very aware that when government funding goes down, our clinical trial system suffers as well. And these are the things that are the important things that are done within the publicly funded trials. So, in summary, we've discussed why research is important and how this occurs through clinical trials. We have also discussed the different phases and what the purpose of each phase is. We've also discussed the different parts of the trial protocol, including the consent form. And patients can speed up this process by entering clinical trials that are right for them and by donating their tissue. Next. Right. So remember to ask if you're eligible for clinical trials and consider your treatment options and what's best for you. I think. Is that the end, Peggy?
Oh. Yes, I think that is, and then we've just got a few disclaimers, and thank you. And Patty,